Okay, so this is after meiosis 1, before meiosis 2. Obviously, we've got two genetically different daughter cells. They do not look like the original parent cell, as you can see. So, yeah. Basically, now it's going to be meiosis 2. We're not going to go back to interphase and do that again because meiosis has 2 consecutive cell divisions. Let's see. So to start it off, we'll go into one cell here. For now, we bring the Twizzler cell back. Go into this guy. Here we go. We have what used to be homologous chromosomes separated from their pairs. It's so sad. I never get to see them due to the law of segregation. But prophase start to condense the this is prophase 2. This is prophase 2. The nuclei start to disappear as it's ready. And I go back. Here come the centrioles. They start to shoot out the meiotic spindle, microtubules, and now this time they're not going to line up in pairs like meiosis 1 in metaphase 1. In metaphase 2, they're just going to line up like they did in mitosis, back to back. We're going to have there's the centromere here, 2 sister chromatids, and then it's going to be pretty much the same as meiosis, or pretty much the same as mitosis. All we're going to see, the motor proteins are going to walk down the microtubules, grab onto the kinetochores, rip them apart, and then boom, there they go to the opposite side of the cell. That's anaphase 2. And obviously you can tell these are not the same. This one looks different than this one. Okay. Yeah, turbo, come on. Okay, so after that anaphase, telophase 2 begins to happen. They start to divide, and cytokinesis splits them apart all the way. Boom, there is 2 separate cells. They are genetically different, but it's not just 2 that get made in this round, because there was another one that was originally there was another one made after meiosis 1. We got to go back to that one. So let me see if I can find it. This is the other cell that was made. It's basically going to do it's 2 cells dividing at the same time. So after meiosis 1, it makes that one that just divided, and this one, they're doing them at the same time. Obviously, prophase starts to get ready. Metaphase 2, they line up in the middle. They are different. They're not they're not lining up in pairs. They're lining up in a straight line. No crossing. There's no crossing over. No synapses. No chiasmata in meiosis 2. It's just the sister chromatids. Oh my gosh. Okay. And yeah, there's no sister chromatids. And then after this is metaphase 2 right here. Obviously microtubules, motor proteins come down, grab them, split them apart, and then boom. That's anaphase 2. Hey, he's eating the chromosomes. Hey, stop it. No. Okay, and then okay. That is telophase 2. So telophase 2 is going to start splitting them. Daughter nuclei start to form, and then boom, that is cytokinesis. 2 more genetically different daughter cells. So if you remember from earlier, these 2 divided. So it's going to look, you're going to have like 4 genetically different daughter cells. So like that's going to be a cell, that's going to be a cell, that's going to be a cell, that's going to be a cell. They're all genetically different. So meiosis is going to produce 4 genetically different daughter cells. So if it's a male, this is a sperm. These are all different sperms. Or if it's a female, these are all different eggs. And they have half the amount of chromosomes, making them haploid cells, and 1/4 the amount of DNA. So yeah, that's the end of mitosis. Or not the end of mitosis, that's the end of meiosis. And having all these genetically different daughter cells will help ensure genetic variation and survival by the because you don't get to choose which sperm. So like if you don't get to choose which sperm goes and fertilizes an egg, so it could be that one, could be that one, could be that one, and they're all gonna shoot out different genetic combinations, which is gonna help us survive.